Today's video is on active transport, but before we look at active transport, it is very important we revise on osmosis and diffusion as part of the ways that substances can move in and out of cells. Osmosis is the transfer of molecules from a region of lower concentration to a region of higher concentration. But in osmosis, it should be noted clearly that the molecules that are being transferred are always water molecules, whereas diffusion is the transfer of molecules, which is usually the molecules of the solute in the concentrated region to a region of lower, from a region of higher concentration to a region of lower concentration. But active transport, on the other hand, is the movement of substances from a region of lower concentration to a region of higher concentration across a living cell membrane using energy from the cell. Diffusion is a natural process. It is natural for molecules to move from a region where they are highly concentrated to a region where they are not much concentrated. That's a lower concentrated region. So in order for molecules to move against nature, that is to move against the concentration gradients, energy must be used, and this is what we call active transport. So active transport is the movement of substances from a region of their lower concentration to a region of their higher concentration across the living cell. The rate of active transport is reduced by lack of oxygen and low temperature. So this means that how fast active transport can occur depends on the amount of oxygen available. If the oxygen available is very low, then active transport will happen slowly. And if the temperature is also low, active transport will also happen slowly. Oxygen is needed in the energy making process called respiration. Therefore, if no oxygen is available, active transport ceases or stops immediately. Examples of active transport are absorption of sugars and amino acids into the blood system from the small intestine in humans. Also, the absorption of some mineral salts from the soil by the roots. This means that particular mineral salt that is being absorbed are of higher concentration inside the root compared to their concentration levels in the soil. So moving from the lower concentration, that is the soil, to the root will require the use of energy to move against the concentration gradient. And this is an example of active transport in plant cells. Endocytosis. Endocytosis is the process by which large molecules, such as proteins, are transported into cells by forming vesicles. The idea of vesicles could be imagined as a bubble in a liquid. The plant cell's cytoplasm, or the cell cytoplasm, could be imagined as a liquid, and a vesicle can be imagined as a bubble inside the cytoplasm. If the material engulfed is quite large and solid in nature, the process is called phagocytosis. If the material engulfed by the cell is liquid or contains dissolved molecules, the endocytosis is referred to as pinocytosis. Pinocytosis is common among the cells of multicellular animals. For example, human egg cells are nursed by surrounding cells that secrete nutrients. The maturing egg cell takes up these nutrients by pinocytosis. If you are familiar with the terms endo and exo, then you will know for sure that the term endo means inside and exo means outside, and it applies with endocytosis and exocytosis as well. Exocytosis is the process by which large molecules are expelled from the cells. So just as endocytosis is how large molecules are taken into the cells, exocytosis simply tells us how large molecules are taken away from the cell, or expelled from the cell. During the process, a cell discharges materials by packing it in vesicles. Remember, I said earlier on that a vesicle can be imagined as a bubble inside a liquid. And moving the vesicle to the surface of the cell, the membrane of the vesicle fuses with the cell membrane and the contents are expelled. It is by the process of exocytosis that plant cells move materials from dictysosomes out of the cytoplasm to build the cell wall. In animals, the secretion of digestive enzymes and hormones is by exocytosis. In this illustration, endocytosis is shown as seeing the large molecules are entering the cell by making vesicles. Number 2 shows how part of the cell membrane forms a vesicle. As I told you, a vesicle could be imagined as a bubble inside the cytoplasm. And this illustration summarizes it all: endocytosis and exocytosis. With endocytosis, the large molecules are entering by forming vesicles, whereas at the tail end, exocytosis, you see the large molecules moving through vesicles. At the end of the cell membrane, the vesicle joins the cell membrane rather than leaving the cell completely.